Hi, I'm James Graham, CEO of Recce Pharmaceuticals Limited. Today, we're talking about beyond existing antibiotics, finding a solution to AMR and sepsis, presented by Sepsis Alliance at the AMR Congress. The usual disclaimer, the nature of these materials. We start with who is Recce and what are we about? Recce Pharmaceuticals, as you may tell by my accent, is an Australian-based biotech company developing a new class of synthetic antibiotics. In fact, it's the most clinically advanced new class of antibiotics in the world at this time, and the most advanced sepsis drug candidate for broad spectrum bacteremia, septic infections. We have multiple preclinical candidates, multiple clinical candidates, and have a qualified infectious disease designation awarded by the US FDA, which provides us 10 years of market exclusivity above and beyond our patent position, plus the addition of a fast-track regulatory review for the life of the regulatory process. As we mentioned, the Pew Charitable Trust affirms us as the most clinically advanced new class of antibiotic and the most clinically advanced synthetic antibiotic in the world at this time, and it is a pleasure to share this with you. So what is this challenge of antimicrobial resistance, or AMR? The challenge when looking at economics is enormous. The cost of AMR is $55 billion each year. $20 billion for healthcare and $35 billion in the loss of productivity. It is estimated to affect 10 million people per year, resulting in death by 2050. And there's barriers to innovation. It has been slow in that there has not been a new class of antibiotics in over 40 years. Those bacteria have continued to mutate. The antibiotics have not kept up. What a terrible history this demonstrates. Where every antibiotic is introduced, an antibiotic resistance is identified. Bacteria have been on the planet for over 3.5 billion years, and roughly 3/4 of antibiotics in development have derived from existing antibiotics. There is no new drugs. So we've tackled that challenge of hypercellular mutation to make the antibiotic introduced cycle extend far beyond our life cycle into the future. So why have existing antibiotics failed? Well, they've got specific mechanisms of action. I think of them a little like a lock and a key. You've got your bacteria and you've got your antibiotic. Today, the two may come together. But for that bacteria to survive, it will mutate and that lock and key will no longer work. The bacteria have evolved and the antibiotics simply have not kept up. Often there's a specific mechanism of action depending on the type of bacterial species as well. Whereas when you look to sepsis, any bacteria in your blood is bad bacteria and you want to get on top of that challenge fast. Who's tackling that challenge? Well, I'm the managing director and CEO, supported by a brilliant team of researchers. Dr. John Prendergast, chairman of a number of New York Stock Exchange biotech companies, with his doctorate in the study of bacteria and blood diseases. Dr. Alan Dunton, former global CEO of Janssen Research, Johnson & Johnson Research as it's commonly known, and has gone from concept to commercialization with a number of antibiotics. In fact, I think even the very last set of antibiotics approved. So what's this problem of sepsis? It's a big problem. It affects around 50 million people worldwide every year. In fact, it kills more people than prostate cancer, breast cancer, and HIV/AIDS combined, resulting in a 1 in 5 fatality rate in the community, as that ratio represents, or a 1 in 3 when we look to hospital. There are no drug therapies specifically approved for the treatment of sepsis. So how does sepsis come about? Sepsis is where you get a break in the skin, whether it be from a skin structure infection, being you've had a break in the skin, be it accidental through an accident or intentional through a basic surgery, and that bacteria gets in and it spreads and multiplies. A common cause is urinary tract infections, an underlying UTI infection, which there's a huge prevalence a drug resistance. In fact, it says here more than 92% of bacteria that cause UTIs are resistant to at least one antibiotic, and almost 80% are resistant to at least two. There's some scary statistics. The challenge we really have is when bacteria enters the blood, it grows and multiplies and it spreads around your body. Every hour left untreated, the likelihood of survival decreases by 8%. So you have to act quickly. But more importantly, you have to act effectively. Existing antibiotics is designed with this type of bacteria and this antibiotic. If you use the wrong type of antibiotic on the wrong bacteria, not only do you contribute to antibiotic resistance, but you often have a very toxic effect on the patient through the overwhelming introduction of the toxic agent. Recce's antibiotic is designed to not care. We don't discern between a gram-positive bacteria, including your hypercellular mutated forms, gram-negative bacteria, including your hypercellular mutated forms. Any bacteria in your blood is bad bacteria, and we're designed to be administered on the first presentation of the patient, not reliant on those time-consuming diagnostics. The present formula or triage is you do a blood draw, you cultivate it out, and you work out two things. One, what type of bacteria is in that patient's blood? And two, if at all, that bacteria is susceptible to an existing antibiotic. Time-consuming guesswork. Recce is to be prescribed at first patient presentation, as our clinical studies have shown. So what is this wondrous new drug? It's synthetic. It's a small molecule polymer. It's soluble at the pHs of the blood to the opposite acidity of the stomach. It's got a mechanism of action that's designed with purpose. It overcomes the hypercellular mutation. It breaks down faster than any other antibiotic. So if there is an inflammatory response, it has no target to hit, so there is no inflammatory response. We work against the broad spectrum nature. We are manufactured through a method of synthesis, not a time-consuming and toxic method of cultivation of all existing antibiotics. And we're designed with purpose, meaning we kill the full suite of escape pathogens and keep on working with repeated use. That hypercellular mutation has always happened and always will, but we continue to work. So we look to the challenge of what is this antibiotic resistance crisis. And as we see here when speaking with global clinicians, they say, we need new antibiotics able to kill drug-resistant bacteria. It is essential to saving modern medicine. That's the Wellcome Trust. The lack of new antibiotics threatens the global efforts to contain drug-resistant infections, World Health Organization. So some quite reputable large organizations screaming for the challenge that is antimicrobial resistance. How are we addressing those challenges? Well, we don't contribute to AMR. And we don't contribute based upon our mechanism of action. We work faster than any other antibiotic to date. We actually appear to take the selective pressure from existing antibiotics fighting us, from the bacteria surviving the existing antibiotics to try to mutate to survive against ours. And we alleviate that selective pressure to ensure that not only our compound works and keeps on working with repeated use, but existing antibiotics may become more effective, as our preclinical studies have shown, and may actually, as a result of that, become more valuable. A win for patients, a win for clinicians, and good for modern medicine. This is actually an example from our very early studies against a number of different bacteria, and we're looking at amoxicillin here, which sells at about $10 billion per annum. After 2 to 4 cycles against E. coli, that's exposure repeat cycles, amoxicillin stops working. After 4 or 6 cycles against Pseudomonas aeruginosa, obviously a nasty, difficult to kill one, they stop working. And 6 or 8 cycles against staph infection there. We work as well the first cycle, second cycle, all the way through to a minimum of 25 repeats. No matter the gram-stained bacteria, no matter the repeated exposure, no matter the defense mechanisms that those bacteria put up, lipid outer layers, biofilms, efflux pumps, all that usual stuff. We work and keep on working with repeated use. We do not succumb to bacterial resistance, which is the fundamental challenge of antimicrobial resistance today. We've informally since taken this out many, many hundreds of times. This 25 cycles represented here was actually from my time as the founding investor of this company, indicating whether this compound had the desired characteristics we wished. This is an example against the full suite of escape pathogens. So escape by the acronym you can see there. What's important here in it is that we work by way of a 6-log reduction in all instances in as little as 1 hour. In fact, I think in some examples there you're seeing as little as 30 minutes. A 6-log reduction is a 99.9999% kill of the underlying bacteria. We don't care what the bacteria is. As long as it's bacteria, we work and keep on working with repeated use. We don't make that claim related to fungal cells because we avoid the commonality between they and healthy eukaryotic cells, which provides a safety effect. Independent research has demonstrated how we work, and we elaborate that upon, by example, there's an abstract that can be clicked in the available presentation here and a number of others available online, where we shut down the ATP synthesis or the beating heart of that bacteria. We shut it down in actively growing cells. We shut it down in stagnant, non-growing cells. They're the more challenging ones to treat. And we shut it down irreversibly. The bacteria can't be brought back to life, and dead is dead, as they say. Efflux pumps don't affect it. There's about 10 to 15 different overt mechanisms which we overcome or we establish. One of them I also like is stopping the ATZ complex strain. Forgive, it is a bit late in Australian time over here. Basically, it means we stop the cell division of the bacteria, or in simple terms, we stop the bacteria spreading and multiplying. This is at a molecular level. So on the left-hand side, you'll see in a dish in real time bacteria, the little ones, they're your deadly enemy. And the healthy cells, the larger ones, they're your ally. So you want to stop the little ones growing, but let the healthy cells continue to grow and multiply like a healthy body would. That's what you see on the right-hand side there. In the same dish, at the same time, the deadly bacterial cells are dead. In fact, if you pick one with your eye, you might even see it disappear, whereas those healthy cells continue to grow and multiply unencumbered. Here it is on a totally opposite gram stain. Naturally, staph is just quite a bit slower growing bacteria. They're typically more difficult to treat. Nevertheless, you see on the left-hand side, the staph and healthy cells growing and multiplying, whilst the bacterial cells, so staph are dead, eukaryotic cells continue to grow and multiply. On the right-hand side, with R327 present, of course, that is exactly what is occurring. So you have a selective mechanism of attack, meaning you can harm your enemy because there's no such thing as good bacteria in your blood, whilst avoiding the healthy common cells in the process. We work faster than any other existing antibiotics, so the time required or exposure required to the human body and system is potentially reduced. So here against E. coli, by example, you've seen your tetracyclines, your ciprofloxacins, colostin, that's pretty well poison. They're not working in a short period of time at all. Overall, there will be some effect in some instances, but you see ours works quickly and works effectively the moment it is introduced to bacteria, as this demonstrates. We've since taken this across to an IV study, basically trying to demonstrate from a safety and tolerability perspective how much compound can be infused into human vein with the interest of overwhelming the bacteria through the vascular system. We demonstrated across 80 persons, 60 of which had our compound, that our compound is safe and well tolerated. There were no significant changes to the EKGs or telemetry. There were no changes to any of the pathology, the organ functions, and similar. At the very, 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 very high concentrations, the worst thing you would get is a bit of irritation around the infusion site. As it mentioned there, our compound does appear to be excreted from the kidney, which passes into the bladder. The benefit of having an active compound appearing in the human bladder is 30% of septic patients have an underlying UTI. They're known as urosepsis. This demonstrates the ability for our compound to work very quickly in non-growing E. coli cells, which of course, non-growing basically means the really deadly resistant bacterial cells that are prevalent in UTI infections. Got a nice dose-dependent concentration to our plasma infusion and a nice dose-dependent increase when we look to the concentration of our compound appearing in human urine, meaning it's gone from the blood where it was infused into human vein, passed through the kidney safely, excreted into the human. That gives the potential to assist the patient from the underlying precursor to sepsis, being urinary tract infections, through to possibly kidney infections to the all-out septic state. It's a really desirable compound profile we're demonstrating here. Of course, it's important to work in the presence of human urine, and in fact, in as little as 15 minutes, you're seeing about a 6 log reduction to E. coli associated with our compound there. We're now expanding that study to a fast infusion, so going from a 1 hour, which has been achieved, through to down to about a 15-minute infusion, addressing patients not just in the advanced hospital setting, but across to the early stage first patient presentation GP or medical setting, where you can get a fast infusion in to stop that infection in its tracks and get on top of that overall bacterial spread. Here we look to the compound administered in a different manner. Now, the reason we bring this into this presentation is looking to the overall challenge of antimicrobial resistance. If you have a topical infection, a break in the skin, a skin structure infection, whether it be a diabetic foot ulcer infection, whether it be from a surgery or an accident, if that bacteria is not brought under control, it will enter the vascular system, which will become sepsis, as high prevalence has shown. So we'd look to it from a topical treatment perspective, and I anticipate these following slides may be a little gruesome. Here we go. So this is called the TGA Special Access Scheme. So the TGA is the Therapeutic Goods Administration. It's Australia's FDA equivalent. And the Special Access Scheme is the also known as Last Resort Scheme of the USA. Basically, we have a number of examples here, all absolutely drug-resistant examples. The first we see there is actually a prosthesis into a human femur, I believe it is. And you see significant bacterial burden, totally drug resistant at time 0. After using our formulation as a gel, so just rubbed onto the surface of the wound, nothing else, just a topical gel. After 3, call it 7 days of single application daily, that person's infection was dried up. There were no signs of redness, and in fact, there was no bacterial present. The next one, down bottom left, we see what is clearly a diabetic foot ulcer infection. Very advanced infection. You can see gangrene and all sorts going on there. After 14 days of treatment, just topically applied, a gel rubbed onto the wound itself, that patient has had a complete eradication of their bacteria. The wound is dry, the wound does not contain any bacteria, and gives the best chance for either a skin graft to be applied, because no skin graft will bind when there's an underlying infection, or a minor surgical procedure to close that wound can take place. You cannot do that when you have an infection. However, of course, had that infection continued to grow and multiply, as this patient was unresponsive to all existing antibiotics, that would have been what is witnessed to an amputation. So that is an excellent opportunity. We can't help that these patients, like many diabetic patients, have lost their cellular regenerative capabilities or their wound healing properties, but it gives the best chance of success for regenerative medicine to take place from there. Top right, we see another diabetic foot ulcer, very, very quick treatment. In fact, from day 0 to day 1, a 24-hour treatment, where you can see a red, raw, really irritated wound go across to a completely dry, no infection prevalent. The next one down is actually a flesh-eating bacteria, so that's kind of your grisly, horrible one. Clearly from day 0 through to day 21, the compound has completely dried up, and through the bacterial swab process identified that what was a gram-positive and gram-negative bacilli bacteria, there is now no bacteria evident. Again, this patient has diabetic challenges, but nevertheless, over time, they would heal. The last one we had evidence here, just for simplicity, is a bone wound infection that's evidenced through the toenail itself. You might see in that left-hand toe there, it's actually got a nice sheen to it. I shouldn't say it's a nice sheen because, of course, that's a biofilm to an E. coli. And after 3 days, that sheen is gone. 7 days, although we don't show it here, the underlying bone infection is entirely cured. That has taken place because of the deep tissue penetration of this compound. We've expanded that into a formal clinical study. The first 5 patients have been completely cured of their multi-drug resistant infections. Patient one, where we say escalated therapy, that's due to the person being a 234 kilogram male with multiple comorbidities. It is unethical for us to prohibit them from not using another antibiotic for another challenge of that patient. So it says escalated therapy because technically, by way of protocol, we had to allow them to use another antibiotic for another challenge they were having. Collectively, they were entirely cured of their infection, as well as all other patients treated. An excellent result. We look to our preclinical landscape as a sign of hope in the fight against antimicrobial resistance. These are just some advanced preclinical candidates that we're assessing for. So Mycobacterium abscessus, that's the bacteria that I believe kills around 50% of cystic fibrosis patients. It really thrives in the mucosal layer of the human lung. And we've demonstrated in animals through respiratory inhalation as well as IV, but I really think respiratory is an area we want to focus on there, a significant ability to work. Pneumonia, of course, as a inhalation method, and then, of course, across to the right-hand side, so sinus infection, so the upper sinal tract. We've demonstrated, of course, an excellent capability in animals and a number of special access scheme patients as well. We manufacture it in Sydney, but we are expanding into the United States. We do actually import material from the United States, so thank you. But we really utilize common materials, water, well, everyone knows that, polyethylene glycol, PEG, 50% of drugs utilize that as a binding agent, and a material called acrolein. So acrolein is from the acrylites or propane. It's an inexpensive, useless waste known for its antimicrobial properties. We polymerize that in an incredibly fast method of manufacture to a compound that's about 750 Daltons in size, soluble from the pHs of the blood to the opposite acidity of the stomach. It takes about an hour to make. We're doing it about 500 doses per hour to GMP standard and specification, and we're supporting all of our Australian, Indonesian, and soon to be United States clinical studies. We have patents to support our compound and claims. This is just by way of a nice verification. Composition of matter patents, so methods of manufacture, methods of curative claims, so ways of curing bacterial infections, preventative claims, ways of preventing bacterial infections, which I think will become a bigger area along the way for us, as well as viral infections. Don't use antibiotics for viruses. This is an anti-infective. We just call it an antibiotic for simplicity in communication. But a virus that invades a cell that has a proteinaceous envelope will be attracted to that cell, and we've shown a great ability to bind to it, interact with it. And they've taken our market exclusivity out to about 2041, so good long tenure ahead. Good news, we have launched a new opportunity through Southeast Asia, Indonesia, about 278 million people there, which provides access across the 10 ASEAN countries. This is actually the largest bilateral agreement between Australia and Indonesia, and it's done for the purposes of launching a clinical study, but I believe to be multiple clinical studies, tackling the global health of antimicrobial resistance. Things like a registrational phase 3 diabetic foot ulcer study, foot infection study, I should say, tuberculosis studies in animals, as well as major unmet medical needs for vast human populations are being studied in Indonesia at this moment, and we very much look forward to reporting back upon. So in summary, this is the world's most advanced new class of antibiotic. Clinically, it's the most advanced synthetic antibiotic studied in the world at this time. We work faster than any other antibiotic known to date, and we work broader than any other antibiotic. We've demonstrated an ability to work against the full suite of escape pathogens, as well as, of course, the full list of those that we could get access to of the World Health Organization. I'd anticipate in the time ahead there may be some activity with the World Health Organization's global antibiotic pipeline. But really, the challenge of tackling the global antibiotic resistance is a challenge that we are tackling head on. And on behalf of Recce Pharmaceuticals, it is a pleasure to present to you today, and we welcome any interest.